In this lecture, we will examine structure, biosynthesis, and function of eicosanoids. Eicosanoids are a class of lipids that include leukotrienes, prostaglandins, and thromboxanes. They are all synthesized from arachidonic acid. They are oxidized derivatives of C20 fatty acids, and arachidonic acid is, of course, C20 fatty acid. Now, essential fatty acids, which were mentioned earlier in one of the previous lectures, are essential because they are needed for synthesis of arachidonic acid and then in turn for biosynthesis of eicosanoids. An example of an essential fatty acid is linoleic acid. And this is formula of arachidonic acid, so you can see further desaturation of linoleic acid leads to arachidonic acid. Arachidonic acid is released from membrane, from cell membrane, and eicosanoids are synthesized in response to inflammation and tissue injury. Leukotrienes are biosynthesized in lipooxygenase pathway. Prostaglandins and thromboxanes are eicosanoids that are synthesized in alternative cyclooxygenase pathway. First, we'll consider leukotrienes. The most important are leukotrienes of the series 4, and some are shown here. Leukotrienes of series 4 have 4 double bonds. 3 of those double bonds are conjugated. There are 5 naturally occurring series of 4 leukotrienes. They are A4, shown here, B4, abbreviations are LTA4, LTB4, then C4, abbreviation LTC4, D4, and E4, again corresponding abbreviations. As you can see, LTC4, LTD4, and LTE4 have cysteine as substituent. So they are also known as cystenyl leukotrienes. There is also another leukotriene, LTF4, which is not shown here. It's also cystenyl leukotriene. It is produced in vitro, but has not been observed in living tissues. Leukotrienes are synthesized in the lipooxygenase pathway, which is active in leukocytes and other immunocompetent cells. Biosynthesis of leukotrienes of series 4 starts by the oxidation of arachidonic acid. Leukotrienes of other series have different precursors. Precursors of 3 and 5 series leukotrienes are eicosatrienoic and eicosapentenoic acids, respectively. Biosynthesis of leukotrienes is accompanied by the synthesis of prostaglandins and histamine, which also act as inflammatory mediators. So first step is in biosynthesis of leukotrienes is release of arachidonic acid from cell membrane, and that is accomplished by phospholipase A2. So arachidonic acid is a component of phospholipids that form cell membrane, and so enzyme phospholipase A2 releases arachidonic acid. Then arachidonic acid is oxidized to arachidonic acid 5-hydroperoxide. The enzyme is 5-lipooxygenase, 5-LO. And that enzyme uses 5-lipooxygenase activating protein, or FLAP, to convert arachidonic acid into highly unstable 5-hydroperoxy eicosapentenoic acid, 5-HPETE. 5-HPETE undergoes spontaneous reduction to 5-hydroxy eicosatetraenoic acid, 5-HETE. The enzyme 5-LO, or 5-lipooxygenase, acts again on 5-HETE, 5-hydroxy eicosatetraenoic acid, and converts it into leukotriene A4. That's an unstable epoxide. And then as this unstable epoxide is further transformed, depending on the nature of the cell, it is converted into leukotrienes of class B or C. In cells that contain LTA4 hydrolase, such as neutrophils and monocytes, LTA is converted into dihydroxy acid leukotriene LTB4. In cells that express LT4 synthase, LTA4 is conjugated with tripeptide glutathione to form LTC4. Outside the cell, LTC4 can lose glutamic acid to produce LTD4, and finally, by loss of glycine, LTE4. All three, LTC4, LTD4, and LTE4, have biological activity that is characteristic of cysteinyl leukotrienes. There are medications that act or that affect biosynthetic pathway of leukotrienes. So, for example, corticosteroids inhibit inflammatory response. They act as anti-inflammatory agents by inhibiting phospholipase A2 and preventing release of arachidonic acid from phospholipids. That completely stops lipooxygenase pathway. On the other hand, interleukin-1 increases phospholipase activity and prostaglandin synthesis. And then there are different classes of anti-leukotriene medications, and some anti-leukotrienes act as biosynthesis inhibitors by inhibiting either 5-lipooxygenase or FLAP enzymes. And then other anti-leukotrienes act by blocking cysteinyl leukotriene receptors. So they don't affect synthesis. Leukotrienes are still synthesized, but anti-leukotrienes block action of those leukotrienes by binding preferentially to receptors and preventing leukotrienes from binding and activating the receptors. Prostaglandins are another important class of eicosanoids. Prostaglandins are found in almost all tissues in low concentrations. They act as secondary messengers and are involved in a wide range of activities, including contraction and relaxation of smooth muscle of the uterus, the cardiovascular system, the intestinal tract, and bronchial tissue. They also regulate gastric acid secretion, they control blood pressure, and suppress blood platelet aggregation. They have very short lifetimes in living tissues, so they have to be constantly synthesized, and steady source of precursors is required. Without the essential fatty acids, those processes may be compromised. Biosynthesis of prostaglandins and related compounds, thromboxanes, starts with arachidonic acid. Arachidonic acid is the precursor for biosynthesis of prostaglandins of series 2. The other two common series of prostaglandins, 1 and 3, will not be covered here. A precursor for biosynthesis of prostaglandins of series 1 is 8,11,14 eicosanoic acid, of course, OZ isomer, it's naturally occurring. And precursor for the biosynthesis of prostaglandins of series 3 is 5, 8, 11, 14, 17 eicosapentenoic acid, EPA. So we are looking at series 2 biosynthesized from arachidonic acid. So in the course of biosynthesis, important enzyme is prostaglandin H2 synthase, because that one catalyzes the committed step in a sequence that leads to conversion of arachidonic acid into prostaglandins, prostacyclins, and thromboxanes. So prostaglandins are biosynthesized same way as leukotrienes, starting by release of arachidonic acid from cell membrane, from phospholipids, by the action of phospholipase A2. And the same way inhibitors like corticosteroids act, they also prevent synthesis of prostaglandins and thromboxanes by preventing release of arachidonic acid from cell membranes by inhibiting phospholipase A2. And of course, conversely, also interleukin-1 increases the activity, phospholipase activity, and increases synthesis of prostaglandins. Now, arachidonic acid is converted by the action of enzyme cyclooxygenase into prostaglandin G2, PGG2. The enzyme is actually PGH2 synthase, which is composed of it has two activities, cyclooxygenase and peroxidase. So first is action of cyclooxygenase to give highly unstable prostaglandin G2, and then peroxidase gives more stable prostaglandin H2, which is precursor for synthesis of various thromboxanes, prostacyclins, and prostaglandins. So from prostaglandin H2, enzyme thromboxane synthase synthesizes thromboxane molecule. And you can see structure of thromboxane molecule. It doesn't have this characteristic ring of prostaglandin. Instead, it has 6-membered ring with an oxygen in it. Then prostacyclin synthase synthesizes prostacyclin compounds. And you can see that prostacyclin has an additional ring, additional cycle. And finally, typical prostaglandins. So from prostaglandin H2, PGD or PGA synthase enzymes respectively synthesize prostaglandins of D series or E series, and E series can be further modified to prostaglandin of F series. Cyclooxygenase enzyme is abbreviated as COX enzyme. Non-steroidal anti-inflammatory drugs, NSAIDs, inhibit cyclooxygenase pathway, so they inhibit synthesis of prostaglandins. But since they only inhibit cyclooxygenase and they don't inhibit lipooxygenase pathway, they don't inhibit synthesis of leukotrienes. So while corticosteroids inhibit synthesis of all eicosanoids, COX inhibitors inhibit only synthesis of prostaglandins and thromboxanes. So they only inhibit cyclooxygenase pathway. There are different types of COX inhibitors. They are classified as COX-1 and COX-2 inhibitors. They are inhibited by non-steroidal anti-inflammatory drugs. And a COX-3 inhibitor is actually inhibited by Tylenol. Tylenol has a little bit different activity. In a course like this, we cannot go into details. COX-2 inhibitors are more selective, but they're also more dangerous, and they're not considered to be any more viable medications. Finally, we will briefly look at anti-inflammatory role of omega-3 fatty acids. Omega-3 fatty acids are well-known anti-inflammatory compounds, anti-inflammatory agents, and they're often used in that role as food supplements. Omega-3 fatty acids are commonly found in seafood and in nuts. They are also readily available as food supplements, and omega-3 fatty acid metabolites are responsible for their health effects, not omega-3 acids themselves, but their metabolites. They appear to play a role in preventing cardiovascular disease and in reducing inflammation. The metabolites are electrophilic oxo derivatives, so-called EPOX derivatives, and are produced by the action of COX-2 on omega-3 fatty acids. Aspirin enhances the effect by causing an increase in production of EPOX or electrophilic oxo derivatives. And this completes our brief investigation of eicosanoids.